First of all, I'm happy to be here to introduce my lab to you, and I hope that because my Chinese is very limited, so I hope that with my ability in English, we can give you something to take home for today. And actually, when I enter the room, then there's a student that asked me a very good question. And why I am in the analytical chemistry group? That my lab name is biomedical chemistry lab. And actually, analytical chemistry is a suburb of chemistry, right? You all know that. But even for analytical chemistry, there's a lot of things you can do with it. So for me, I'm focused on the biomedical application using analytical chemistry. So that's why I named my lab is biomedical chemistry lab. And a little bit about myself. Okay, so I come from Vietnam. So I think now all of you are Taiwanese, so I'm hoping that maybe later I can see some Vietnamese face here. And I come to Taiwan in 2016 to study my master degree and then continue for PhD and then postdoc. And then I'm also honored to be here in our department, the Department of Chemistry of NTNU. So for the research topic, actually, I would like to focus on more on the bioanalytical chemistry. As I just mentioned to you that I would like to focus on the biomedical application. So I'm more like an application guy. So for example, I like to do biomarker detection. I think you may know what is biomarker, right? So biomarker is a substance or could be a protein or could be anything that relates particularly to a disease. So if, for example, if we have that substance, we have that protein in our blood, then we can conclude, we can predict, or we can diagnose that we got that disease. For example, I do infectious disease biomarkers. For example, dengue virus. You know dengue fever, right? Tăng
Yes.
Beside that, my research focus is on to develop the methods for those biomarker detection. So, for example, when you go to the hospital to take your blood, so then the doctor or the technician to do some tests and then finally give you some number. So from until you take your blood, from you take your blood until you get the number, there's a lot of things between. So I focus on how to do that or and how to improve that part. So the material I'm using is fluorescent nano diamond, and I will introduce to you a little bit about fluorescent nano diamond later. And using some of the method, for example, paper-based assay, or people calling lateral flow immunoassay. So is there any one of you know what is lateral flow immunoassay is? No? I think you will know it when I show it. So I will show it later also. And another application I would like to focus on the polymer chemistry for biomedical application, such as drug delivery. I think let's go forward. You know this one, right?
Yes.
Yeah, the rapid test kit.
Yes.
I think a lot of you have been used it using during the COVID-19, right? So this is one type of lateral flow immunoassay. So lateral flow immunoassay is a more scientific way to call it, right? So, but we just usually call it rapid test. So here, the two lines you see here, usually anyone know what is it made of? Why we see the color?
Uh, because the combination between a substance and
Okay, so usually that color is made from gold nanoparticle, gold, or polymer nanoparticle that has a color. So we can see the color. So the principle is on the strip, before you put your sample on, there's already has some antibody on it. And then if in your blood have the biomarker have for sample have the COVID-19 antigen, it's gonna be captured by the antibody on the line here, and then it will also, and then it will capture the gold nanoparticle, so you can see two lines. So for me, instead of using gold nanoparticle, I replace the gold nanoparticle with nanodiamond because the property of nanodiamond is have something interesting. For example, if we call it fluorescent nanodiamond, you know what the fluorescent means, right? In what? Okay. So it means that it can emit a light when we excite it with some other light, right? So for fluorescent nanodiamond, usually if we use a green light and point to it, it can emit a red light. But if that's the only property, then it's quite similar to other things, right? Another property is that fluorescent intensity will change when we put a magnetic field on it. So, for example, here you can see this is the fluorescent intensity, the spectrum. So all of the light that emit from the fluorescent nanoparticle in the water. So in here, we also have the water fluorescent. So if we would like to detect the fluorescent from NAD only, it's very challenging because it's overlap. But when we apply a magnetic field and then and do some mathematic calculation, then we can filter out the fluorescent signal of the nanodiamond only. So it means that if we are using this kind of particle, we can have something like background free detection, which is it doesn't depend on the background. It doesn't depend on the signal come from the membrane, for example, or come from other substance in your sample, right? So so that's why that we think it should be more sensitive compared to the one that you are used to use. Okay, so to apply it, this is just the diagram of the instrument that we are using, but I think maybe it's not important today. Then this is the principle of what I just mentioned to you for lateral flow immunoassay or immunoassay in general. So immunoassay have two parts, right? The first part is immuno. Immuno is the interaction between antibody and antigen, right? So that's what we call immunocomplex. So for an assay that view that interaction, we call immunoassay. So usually, for example, if we have, let's see this one sandwich is the most common one. If we usually we're gonna have one antibody on the strip like this. Similarly, in your rapid test, and then when we flow the sample, the target antigen gonna be captured, and also the nanoparticle with another antibody will be captured. So it means that when we have no target antigen, we have no signal because there's no binding here, right? If we have the target antigen, we're gonna have signal. So that's the difference or the principle of how can we detect things. But I'm more care about the sensitivity, or it means the limit of the detection. For example, if we are using gold nanoparticle like the one that we are usually use during COVID, the detection limit is very limited, like me. It's not too long. So we will apply the different type of material. So we would like to make it more sensitive. So that's why we use fluorescent nanodiamond. And there's just some number for you to take a look at it. And So far, we have been tried with so many biomarkers to get its detection limit. So, for example, the detection limit now, the best one is about 10 picogram per milliliter. So, do you have any idea 10 picogram is what?
Nano.
Nano is 10 to minus 9, right?
Yes.
How about picogram?
12.
Minus 12, right? So this means that we can detect something if it's only 10 to minus 12 gram per milliliter, which is very sensitive, right? Okay. So that is one type of immunoassay or one type of the method that we develop. And there's another type of method that we also develop in our lab, also using nanodiamond. So you can see this assay called turbidimetric immunoassay. So it means that if you have your antibody and then you also have the antigen, the target analyte together in a solution. If you form an aggregate, it will aggregate each other. Because it aggregates each other, so the stability change or the absorption of the solution change. So based on this, we could calculate the amount or the concentration of the target analyte. So instead of this, we are putting a nanoparticle inside. So it's gonna enhance because normally when we don't have nanoparticle, so in order to see the change of the turbidity, it's gonna need a lot of energy, a lot of time analyze. But if we have a nanoparticle, so the nanoparticle is gonna act like a cluster, right? So when it aggregates together, it's gonna be easy for us to see the change, which means that you're gonna have higher sensitivity compared to without a nanoparticle. So this is the line. For example, if we have more target analyze, the shorten the solution gonna increase because they aggregate each other. Okay. Okay. So this just to show you the kinetics of the assay. Just so it means that you can see here. This is the time that we put the sample inside until the time that we can see the change in the signal or the change in the absorption. So very fast, like within 5 minutes, you can get the result. And some TM, some supporting data to show the aggregation of nanoparticle when we have the target analyte. So all of these are using the interaction between antibody and antigen, so that we call immunoassay. And some of the number, for example, the calibration curve, the stability of the assay, for example. So another thing that we are doing in our lab is that we do drug delivery. So drug delivery means that you have a drug, for example, anti-cancer drug. You would like to deliver it into the cancer cell or the tumor site only, not the whole body. So that's when we need a drug delivery system. Okay, so I specialize in synthesize polymer with different type of response. It means that, for example, in the cancer microenvironment, we have the pH is slightly more acidic compared to the normal uh, site. So for example, in normal condition, your normal tissue have a pH around 7.4, but in the tumor environment, we have a pH around 6.2. So if we could have something or a particle that can be broken or respond to more acidic pH, it means that that particle will respond to that tumor environment, right? So if you have anti-cancer drug inside a particle, so it will release only at the tumor site. Okay.
It will be target specific.
Not yet, not that target specific yet. So this one called control release. So we control the release. But for targeting, we're gonna need something else, like For example, in the tumor side, we're gonna, for example, in tumor, some tumor have more type of receptor compared to the other receptor. So we're gonna use a substance that target to that receptor so that we can call targeted. But this type is called control release, not yet target. But but it's possible to combine two things together, right? So if whenever we are using polymer, then it's easier for us to control because we can synthesize the polymer the way we want. For example, if we would like to put the targeting thing inside, we can just conjugate it together. If we would like to put the control release inside, we can just conjugate together. Okay. So for example, so this one is a molecule. This one is hyaluronic acid, the one you are usually seeing in cosmetic. I think you are uses a lot for uh, to moisturize. Okay. Hyaluronic acid, HA. So it's a natural polymer and it's hydrophilic because it dissolves in water. And then we have another substance, this one called ESQ3. ESQ3. Or the full name is black hole quencher, but we don't use that quencher property. So I just call it ESQ3. In this one, they have what is this bonding called? End to end, what it is? Azo, right? Yeah, azo bonding. So this type of bonding very sensitive to pH. So, for example, if we have, for example, acidic pH or another condition, it will be broken. It will be break. Actually, the property of the tumor that we are using here is that the low concentration of oxygen. Because in tumor, they grow too fast, right? So they consume a lot of nutrients, they consume a lot of oxygen. So the oxygen concentration is lower compared to the normal cell, right? So these azobonding will be uh, reduced by, no, will be break by under the low concentration of oxygen. So when we have this bonding inside, and this one is hydrophilic, this one is hydrophobic, and so because we have two things, hydrophilic, hydrophobic, and then so it can assemble become nanoparticle because then the hydrophilic part will stay outside, the hydrophobic part will stay inside. Then it's become nanoparticle. Once we have a nanoparticle, we can have the drug, anti-cancer drug. For example, here I have EOX, is doxorubicin, an anti-cancer, to lock inside that particle, right? So under specific condition, for example, low oxygen concentration condition, the particle can be break due to the chain of this one, and then the drug can release out. Another property of the HA is that it can target the CD44 receptor on the tumor, which is this one has more on the tumor, on the cancer cell compared to the normal cell. So it seems like we're gonna have like control release by this, and then the targeted release by this. So this is just a principle of what we would like, what we are doing. And then another design, for example, we also make a polymer because like with polymer we can have a lot of things in it. So we have this one is hydrophilic polymer, this one is hydrophobic polymer. The reason we would like to make it hydrophilic and hydrophobic is that it can assemble into particle, nanoparticle. So the thing that with hydrophilic property and hydrophobic property, we call it amphiphilic property. So between them, I put a linker. So this linker gonna be break under the ROS. So ROS is the reactive oxygen species, so which also have high concentration in the cancer. So, like you see, if we would like to target the delivery to the cancer, we need to know the property of the cancer cell or the tumor environment. So then we utilize it for the delivery or for our purpose. So that's it. And another one is also using polymer because with polymer I can do a lot of things. And this kind of polymer can specifically coordinate to aluminum. So we are using the polymer to detect the concentration of aluminum. Okay, and that's it. So my main focus is on the bioanalytical chemistry with application in biomarker detection. But besides, I also do smart polymer area and polymer based sensor. So I think this is the uh, collaborator and the funding. This is thank you.